Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So hello guys, so welcome to my session. So thank you so much for joining my session. So please do like, share and subscribe. So for all those who are new to my session, so Monday, Wednesday and Friday I teach for a class 12 chapter, and Tuesday, Thursday, Saturday I teach a class 11 topic. So today is Wednesday, so we are here talking about a class 12 topic, that is sexual reproduction in flowering plants. So this is lecture 3 of your sexual reproduction in flowering plants. I hope you remember the first 2 lectures. Yes, so if not, no worries. So we will have a quick recap of them. Yes, Anthony Abhi, very good evening. So thank you Anthony Abhi for joining my session. So please do like, share and subscribe. Yes, so do you have any doubts in the previous 2 lectures? So things are clear? Yes, so shall we move forward? Yes? Alright, so let me go into the topic, that is sexual reproduction in flowering plants. So this is lecture 3 of your sexual reproduction in flowering plants. So what are the topics that we were discussing so long? So we were talking about introduction, so how flowers are closely attached with human welfare activities, so how we use flowers from day-to-day activities, and what are the 4 worlds of a flower, so calyx, corolla, androecium and gynecium, right? Then we spoke about the pre-fertilization structure and events. Yes, Vinu, hi, a very happy good evening Vinu. So thank you Vinu for joining my session. So please do like, share and subscribe. Okay, so what are the pre-fertilization structure and events? So for the fertilization to happen, you need gamete, right? So for the gamete to be produced, so you need to talk about the structure of the male and the female reproductive organs. So pre-fertilization structure includes the description of your stamens, microsporangium and your pollen grains, and in case of female, it is your pistil, megasporangium and your embryo sac. Alright, so then today we'll talk about your pollination, because in the previous lecture we have completed till your pistil, megasporangium and your embryo sac, right? So now we will talk about pollination, and then to some extent about double fertilization. I guess double fertilization we can start off in the next class. Alright, so that is on Friday, okay, and then we will talk about post-fertilization structure and events. So after fertilization happens, so we'll talk about the development of your endosperm, your embryo, your seed and apomixis, and what is polyembryony. Alright, yes? So ma'am, please start physics and chemistry classes. Alright, so once again I'll let the management know. Okay, so please give us some time before we sort out and start classes here. Okay, fine. Then, so talking a quick recap about stamen, microsporangium and pollen grains. Come on, quickly tell me the order. So quickly let's revise what is your stamen, microsporangium and your pollen grains. So stamen has 2 parts. What are the 2 parts of your stamen? So it has 2 parts. One is your anther, the other one is your filament, right? Yes? So it is like this. So this is your filament and this is your anther. So your anther is bilobed and it is also diathecous. So what do I mean by diathecous? So it has 2 wall structure. So that is why it is known as diathecous, right? So your pollen grain is also a cornered structure. It has 4 microsporangia. So what is this microsporangia? So microsporangia develops into your pollen sac, which in turn produces your pollen grain, right? Yes, hi Krishna Kumar, hello. So thank you so much for joining my session. So please do like, share and subscribe. So microsporangium is the pollen sac wall. Yes, you're absolutely right. So what is the ploidy? So microsporangia is 2n. So pollen sac is 2n. So pollen grains will be haploid. Alright? Yes? So when the anther is young, the central region of your microsporangia consists of a group of homogeneously arranged tissue which is known as sporogenous tissues. Right? So these sporogenous tissue houses your pollen mother cells or your spore mother cell. Right? So this undergoes meiosis to produce 4 microspore tetrads. So these microspores develop into your pollen grains. So before that, the wall of the microsporangia is made up of, the wall of the microsporangia is known as microsporangium. So microsporangium is also diploid and it has 4 layers. So what are the 4 layers? Epithelium, endothelium, middle layer and tapetum. The first 3 layers have the same function of giving protection and causing the dehiscence of anther from the pollen grains. Sorry, dehiscence of pollen grains from the anther. Alright. Then the last layer, that is tapetum. So tapetum helps nourishing the developing pollen grains, and the cells of tapetum are, they are having a dense cytoplasm plus nucleus, and they are having more than one nucleus. So it follows endomitosis, where your karyokinesis is not followed by your cytokinesis. Alright, yes, very good Antonia B. Yes, Harini, a very, very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. Alright, clear? Okay, so as the anther matures, so these tetrads, they move from each other, and each of the microspore develops into a pollen grain. Alright, yes. So now talking about the microspore. So the microspore develops into a pollen grain as the anther matures. Now we'll talk about the pollen grain structure. So pollen grains are haploid because this is your microspore, right? So pollen grain is a double wall structure. The outer wall is known as XII. The inner wall is known as intine. So the outer wall is made up of a substance known as sporopollenin, so which is very resistant to any type of destruction. So it can resist organic acids, alkaline, high temperature, etc. So on your XII, you have certain apertures which are known as germ pores. So in your germ pores, your sporopollenin is absent. Alright, so the innermost structure of your pollen grain is your intine, and intine is a thin continuous layer which is made up of your cellulose and your pectin. Fine, so this is about the structure of your pollen grain. So Arshita, alright, so good evening. Yes, good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay, so I am talking about sexual reproduction in flowering plants. So we are already done with your pollen grains and all. So I'm just giving you a recap. So if you do not understand, stop me right there. Alright, fine. So now talking further about your pollen grain. So when the pollen grain is growing, so when the pollen grain is mature, it consists of 2 cells within itself. So what are the 2 cells of the pollen grains? One is your vegetative cell, the other one is your generative cell. Alright, so pollen grain itself is haploid, so these 2 cells will also be haploid. So pollen grain undergoes mitosis to form your vegetative cell and your generative cell. Your vegetative cell is big enough and it has an irregularly shaped nucleus, and in the cytoplasm of the vegetative cell you have your generative cell. When your pollen grain comes out of the anther at this stage, this is called as a 2-cell stage. Alright, sometimes what happens, your generative cell undergoes mitotic division to form 2 male gametes, so each is a haploid. So thus you have 3 cells, and when your pollen grains are shed at this stage, it is known as your 3-cell stage. Alright, so then we started talking about the pollen allergy that is present, that is causing your bronchial infections, and then we went on to talk about the viability of your pollen grains. So what is the viability of pollen grains? Yes, pollen gets released, that is fine, but the destination of your pollen grains is the stigma of your flower, right? So the moment it reaches the stigma, or the duration that it takes to reach the stigma, should ensure that the pollen grain is live and it is functional, right? So in case of some cereals like your maize and wheat, the viability is just 30 minutes, and in case of families like your rosaceae, the viability is for a longer period, say for 1 year. Yes, of course, you can preserve your pollen grains in liquid nitrogen in -196 degrees Celsius. So just like your seed banks, you have your pollen banks. Alright, is this much clear? So moving forward to the next one, that is your pistil, megasporangium and your embryo sac. So what are the three structures of your gynecium? So you have your stigma, style and ovary, right? So that is your stigma, style, and the basal portion is your ovary. Inside your ovary, you have the ovarian surface. So this ovarian surface is called as the locule. So inside the ovarian surface, you have a cushion-like structure that is known as placenta, onto which your ovules are attached. Fine? Clear? Okay. So the ovule is attached to the placenta by a stalk, okay? That stalk is known as funicle. So the body of the ovule attaches or fuses with the funicle at a particular point that is known as hilum. Alright, so here I have my ovule, alright? Yes, so here I have my stalk, so this is my funicle. So the body of the ovule is attached like this, so this is my hilum. Fine. So the ovule is surrounded by one or two envelopes. Yes, Pallavi Rao, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Alright. So the ovule here is surrounded by one or two envelope structures. So this is the ovule. Alright. What is called as the interflowering period? So that is called as the mature phase. It is not the juvenile phase. So in juvenile phase, you will not have any flowering taking place. Alright. So when biomolecules class will continue, so biomolecules class I will continue on Saturday. Alright, because we are only left with your human here in your neural control and coordination, so which we'll talk about on Thursday. So biomolecules class we'll have it on Saturday. Alright, yes sir, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. So the ovule is surrounded by one or two outer envelopes. So this outer envelope is known as outer integument. And this one is known as the inner integument. So the integument surrounds the ovule at all portion except at one point, right? So here it is not covered. So this opening is called as the micropylar end. So exactly opposite to the micropylar end, this end is called as the chalazial end of the ovary. So this is the basal portion. So inside the integument, you also have another layer that is called as nucellus. So I hope you remember these terms well. So what is a nucellus, what is an integument, what is a micropylar end and what is a chalazial end, right? Yes? Okay, so I have the figure, but on top of the figure I started writing. So just give me a minute, I will clean it up so that we can see the structure properly. So the chalazial end, so it is exactly opposite to the micropylar end. So you have outer integument, inner integument, nucellus, and inside this you have your ovule. Your ovule develops into your embryo sac. Alright, so moving to the next step. So what happens? The cells of the nucellus, so cells of the nucellus are 2N and this has your megaspore mother cell. Okay, so this megaspore mother cell will now undergo mitotic division to form 4 megaspores. So your megaspores are haploid here. So among the 4 megaspores, only 1 megaspore is functional. Alright. As after flowering, again it will start developing and it has to undergo juvenile phase. Okay, so interflowering period, in case of which plants are you talking about? So interflowering is when, interflowering is actually, you can say, the mid phase between your juvenile phase and your mature phase. You cannot call it entirely juvenile phase, because in juvenile phase you do not have flowering, but you cannot call it as an entirely mature phase because it is interflowering period where the flowers are not matured enough to undergo pollination. So it is in between both. So that is why I said it is a mature phase. But what are the options given? So based on the options, we'll have to justify your question. Okay, so can you give me what are the options that is given, so that I will give you the justification as to why it is mature phase or why it is juvenile phase. Okay, fine. It produces 4 megaspores. Unlike your 4 microspores, only 1 megaspore is functional. Only 1 megaspore is functional. Alright, so this 1 megaspore which is formed by this process, so this process is called as megasporogenesis. So this 1 megaspore undergoes mitotic division to form your 2 cells. Okay, it forms your 2 cell stage. So this further undergoes mitotic division to form the 4 cell stage, and this further undergoes mitotic division to form the 8 cell stage. Fine. So in this 8 cell, out of this 8 cell, only 6 cells have the formation of cell wall around it. So in this 6 cells, so 3 cells are present towards the micropylar region and 3 cells towards the chalazial region. The 2 nuclei are present in the center. So the 2 nuclei that is present in the center is called as the polar nuclei. Fine. So the 3 cells that has moved towards the micropylar end develops into the egg apparatus. So the 3 cells that are present in the micropylar end, so 3 cells, so say for example this is my micropylar end. So this is together called as egg apparatus. So it has 1 egg cell and it has 2 synergids. Alright, so this is towards the micropylar end. How many meiosis and mitosis are required in both? Only 1 meiosis. So only the mother cell will undergo meiosis. So mitosis will happen 3 times so that it ends up in a 8 cell stage. Okay, so the mother cell will undergo meiosis to form the megaspore or the microspore, after which only mitosis will happen. Okay, so the mother cell will undergo meiosis to form the megaspore or the microspore, and then the microspore or the megaspore will undergo repeated mitosis. Okay, in case of your pollen grain, it is only 2. Okay, vegetative cell, generative cell, and then generative cell undergoes second mitotic division to form your 2 male gametes. Here it is 3 meiotic divisions. Okay, sorry, mitotic divisions. So this is 2 synergids. Then you have 2 polar nuclei already present. Then towards the end also you have 3 cells like this. So these are your antipodes. And this is towards your chalazial end. So this entire thing is wrapped up in 1 cell wall. So thus a embryo sac, a mature embryo sac is 7-celled and it is 8 nuclei. Sorry, it is 7-celled and it consists of 8 nuclei. So 1, 2, 3, 4, 5, 6, 7, 8. So it is 8 nuclei. But why it is 7-celled? Oh, the 6 cells are walled, but these 2 nuclei are present in this entire cell. So a mature embryo sac is 7-celled and 8 nuclei. Alright, is this much clear? So this is what we were talking about, the functional embryo sac. So your synergids also leads to the formation of your filiform apparatus. So your filiform apparatus helps in guiding the pollen tube inside the embryo sac. Okay, so now the next thing is pollination. So what is pollination? So what is pollination? So before we start talking about pollination, let me explain once again. So we have it like this. The events of sexual reproduction is divided like this. Pre-fertilization. So fertilization, and then you have post-fertilization, right? Fine. So fertilization is fusion of male and female gametes. So it leads to the formation of zygote. So this is your fertilization, alright. So what is pre-fertilization? So you have to form the male and female gametes, right? So here our male and female gametes are formed, that is gametogenesis. Generally it is called as gametogenesis. The formation of male gamete is called as microsporogenesis here. And here it is called as megasporogenesis. Right? So thus our male and female gametes are ready. So your male gamete is your pollen grain and the female gamete is your embryo sac, which is 8 nuclei and 7 cells, right? So all these are ready. Now the next step here in this process is, how will the pollen grains reach the embryo sac? Yes ma'am, good evening, a very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. So this pollen grain has to reach the embryo sac. If you remember, when I was talking about reproduction in organisms, so under that sexual reproduction, I said both the plant gametes are non-motile. The pollen grains cannot move on its own. The embryo sac cannot move on its own. But this has to go into the female sac. That is, they both have to meet, only then fertilization can occur. So what ensures that the pollen grain lands on the female reproductive part? So there should be some phenomena or some method by which your pollen grains land on your embryo sac, right? So that process is known as pollination. So the process by which your pollen grains land on the stigma of your pistil is called as pollination. So that is how your pollen grains will reach the female gamete, and thus the process of fertilization will take place. Understand? So now so long we were talking about microsporogenesis and megasporogenesis. Now we are going to talk about how your pollen grains will reach your embryo sac. So that is with the help of a phenomenon known as pollination. Alright, so shall I move forward? Okay. So what are the different types of pollination? So depending upon the source of pollen, pollination can be of 3 types. The first type of pollination is autogamy, the second type is gatenogamy, and the third type is xenogamy. Alright, so we will talk about each of them. So what is autogamy? So pollination within the same flower. So when a particular flower has both androecium and gynecim, that is when a particular flower is bisexual. So only then pollination is possible. A bisexual flower will have your stamen as well as your pistil. Right? So that is a bisexual flower. A stamen consists of your filament and anther. So your anther consists of sporogenous tissue which will produce your pollen grains. Your pistil consists of your stigma, style and ovary. And inside your ovary you have your ovule or your embryo sac. Right? So when pollination happens within the same flower and the flower consists of both the stamen and pistil, that is the flower is bisexual, then the type of pollination is called as autogamy. Alright, so the transfer of pollen grains to the stigma of the same flower is called as autogamy. So normal flowers, they open, so if it is a normal flower, when I am saying autogamy is within the same flower, right? So the flower should open, the pollen grains are produced and the stigma is nearby, so they land. But this condition is very rare. When a bisexual flower is undergoing autogamy, it rarely opens out. It is in the bud stage itself. So that pollination is easier. Alright? So in this case, when your autogamy is happening, that is pollination within the same flower is happening between the pollen grains of the flower and the stigma of the same flower. So what will happen? There should be synchrony. So the pollen grains should be released at the same time the stigma is ready. The stigma should be ready to receive them. Right? So there should be synchrony between your pollen grain release and the stigma being receptive for the same. Alright, so that is called as autogamy. So anthers and stigma should also lie close to each other. So for this condition of autogamy, so there are certain criteria which has to be met. So the first criteria is that the pollen grains and the stigma has to be synchronized. So they should be receptive, they should get mature at the same time. The pollen grains should mature at the same time. The stigma is mature enough to take up the pollen grains. So this is condition number 1. So condition number 2 is anther and stigma should lie close to each other. Thus pollination is easily possible. Okay. So what are the examples of plants undergoing autogamy? It is the viola, common pansy, oxalis and camellia. They produce 2 types of flowers. Okay. So this is chasmogamous. So the flowers are of 2 types. So all are bisexual flowers. So what is this condition? So flowers are produced in the chasmogamous condition. So meaning the anther and the stigma are well exposed and then pollination happens within the same flower. So that is known as chasmogamous condition. So we are talking about the types of pollination, and the first type of pollination is your autogamy. Alright, so in autogamy, pollination happens within the same flower. So within the pollen grain of the flower and the stigma of the same flower. But for that to happen, either the flower can be in an open state, that is the chasmogamous state, when it is exposed, when the anthers are visible, when the stigma is visible. So that is the chasmogamous condition. The next one is your Gatenogamy condition. So the flower does not open at all. It is present in the bud condition itself. So that is called as Gatenogamy condition. So in the Gatenogamy condition, when it is in the bud state, the anthers and stigma lie close to each other. Thus the anther wall opens, the pollen grain comes out in the bud stage and it is quickly transferred to the stigma. So this is known as your proper autogamous condition. So this is invariably autogamous because this is exclusively, there is no chance of any other pollination happening, right? There is no chance of cross-pollination happening. So you have an assured set of seed produced even in the absence of pollinators, right? So the process of pollination is picking up the pollen grains and landing it on the stigma, right? So sometimes it is through the help of agents, but without the agent also autogamy is completely possible. Fine? So the next step of pollination is your Gatenogamy. So what is Gatenogamy? That is transfer of pollen grains to the stigma of another flower of the same plant. Okay, so if I am having a plant like this, so if I have a flower, so this can be a pistillate flower, meaning it contains only the Gynecim portion, alright, and here I can have a flower which can only contain the stamen portion, or it is the staminate. So when pollination happens between these 2 flowers of the same plant, it is known as your Gatenogamy. Fine? So this is ideally in the same plant, but between 2 different flowers of the same plant. Yes, hello, hi Sathinder. So good evening. So today you are pretty late to the class, but still thank you so much for joining my session. So please do like, share and subscribe. Alright, so this is called as Gatenogamy. So they function basically as cross-pollinators because they need pollinating agents. From this flower to this flower also the pollen grain has to be transferred, right? So here you need pollinating agents, but it is genetically similar to autogamy because it is ultimately the same plant. The pollen from the same plant, the embryo sac from the same plant. So it is also a type of autogamy, but it involves your pollinating agents. Alright, so the next type or the last type of pollination is your Xenogamy. So what is this Xenogamy? So it's the transfer of pollen grains from the stigma, from the anther to the stigma of a different plant. So this is entirely 2 different plants. So in case of papaya, papaya has unisexual flowers. So one male plant and one female plant. If pollination happens between two different plants, it is known as Xenogamy. Alright. So now you understood what is autogamy, Gatenogamy and Xenogamy. Yes. So this brings genetically different types of pollen grains. Okay, now we are done with the types of pollinations. Now we are going to talk about the agents of pollination. So who are the agents who will help us in pollinating the pollen grains? So shall we look into the list of agents? So till this much it's clear, Anthony B. So all those who are watching, Vinu, Sathinder, is this much clear? So we are done with the male and the female reproductive part. So we understood how your male gamete and your female gamete is produced, and then we entered into what is called as pollination. So under pollination we are talking about the different types, that is autogamy, Gatenogamy and Xenogamy. So now shall we move into the agents of pollination? Yes. So no doubt I don't have to repeat anything. Clear? Alright, very good. So moving forward, the agents of pollination, talking about the agents of pollination. So you have two types of agent. One is abiotic agent, the other one is your biotic agent. So abiotic is non-living, biotic is living. So the abiotic agents are wind and water. Biotic agents are animals, and mostly plants prefer your biotic agents. So majority of flowering plants prefer your biotic agents for pollination. Okay. So pollen grains present on the, pollen grains have to reach the stigma, and this is a chance factor in case of your wind and water. So wind and water goes at a very great speed, and if at all the pollen grain is smart enough, it is carried through the wind, right? So but there are high chances that the pollen grain is not properly carried by the wind and it does not reach the exact destination. So that is why, to compensate this chance factor or probability, a large number of pollen grains are being produced. Okay. I completed genetics. When did I? Okay, yes, in YouTube, yes, I have completed genetics. So I have taught principles of inheritance, molecular basis of inheritance, evolution as well. Then I have revised the topics twice. So don't worry. So once I am done with the syllabus, once again I will start the revision. But you can watch my videos. In every video of genetics, I would have solved questions. Alright, Antonia B? Fine. So the pollen, so to avoid this chance factor or probability, so you have large number of pollen grains being produced. Fine. So pollination by wind, the first one, so this is the most common type. So this is known as animalophily. So that is pollination by wind. So pollen grains, so for the pollination by wind, pollen grains have to follow some criteria. So the pollen grains have to be light and non-sticky. That is how it can be carried by the wind. If it's sticky, just stick to the surface. How can it move by the wind? Right? So it has to be light and non-sticky so that it can be transported by your wind currents. And it should also have well exposed stamens so that the pollen grains can easily be dispersed. The pollen grain should be well exposed in air so that when the breeze flows, it can easily move from one plant to the other plant. Your graduation. Yes, I am done with my graduation. Yes, of course. Yes, my graduation in genetics. Okay, so the stigma, how should the stigma be? The stigma should be feathery and sticky so that it can easily capture your pollen grains. Alright, yes, fine. So the flower should have a single ovule in each ovary and there should be numerous flowers present, so in the form of an inflorescence. Alright, so example of your pollination by wind is your corn cob, and it has tassels which is called which is basically your stigma and your style. So once again, pollination by wind has some certain criteria for your pollen grains as well as to your stigma. Your pollen grains have to be light and non-sticky and it has to be transported by the wind currents. Your stamens should be well exposed so that it can be easily dispersed. Alright, so it should have a large and feathery stigma so that it can easily trap your pollen grains. Then your flower should have a single ovule present inside a single ovary, and there should be numerous flowers of the same type in the form of an inflorescence. Alright, so example of your anemophyly is your corn cob. So this is also common in case of your grasses. Now talking about pollination by water. So this is the next abiotic pollinator present. So pollination by water is comparatively rare when we talk about pollination by wind. So pollination, so when a plant chooses abiotic pollinating agent, they prefer wind than water. Okay, because not all are aquatic plants, so not all plants are accessible to water as well, right? So only water pollination is limited only through 30 genera, and it is mostly in case of your monocots. Of course, wind pollination, I told you, it happens in your grasses. So apart from grasses, other monocots, they prefer water pollination. So, and if you also remember, water is the mode of transport for male gametes in case of your algae, bryophytes and pteridophytes. So where did we talk about algae, bryophytes and pteridophytes? In case of your plant kingdom, right? So algae, bryophytes and pteridophytes, the male gamete is released into water. So the water carries them to the female tract, right? That is also there. So example of your water pollinated plants are your Vallisneria and Hydrilla, which are freshwater plants and they need water for pollination. So you also have marine grasses like your Zostria. So they also prefer water pollination. Okay, so not all aquatic plants use water pollination. So when I say water pollination, logically what comes to a brain? So all the aquatic plants will need water pollination because they are nearby water or they are in water, right? But it's not necessarily, not all aquatic plants use water pollination. So water hyacinth and water lily, the flowers emerge above the level of water, and here pollination happens by either insects or by wind. They do not pollinate by water. So once again, not all aquatic plants use water pollination. So the water hyacinth and water lily, they are aquatic plants and the flower rises above the surface of water by a small stalk and it is pollinated by insect or wind. So this is your water lily. So this is your water surface. So it is well above the water surface because it is connected by a stalk. So now pollination happens by your wind or by your insects. Fine. So talking further about your Vallisneria, which requires water pollination. So the female flower reaches the surface of water by a long stalk, alright, and the male flowers or pollen grains are released on the surface of water. So water pollination is of two types. Pay attention here. One is called as epihydrophyly. So water pollination is known as hydrophyly. The other one is hypohydrophyly. Epi means pollination above the surface of water. This is inside the water. Alright, so water pollination is of two types, epi and hydro. So epi is above the surface and hydro is inside the surface of water. So in case of Vallisneria, when I say the female flower reaches the surface of water and male pollen grains are released on the surface, so it is an example of your epihydrophyly. So the male gametes are passively transported by your water currents. Alright, so some male gametes eventually reach the surface of your water. So this is your Vallisneria, alright. Sorry, this is your sea grasses where the fertilization happens where the pollination happens inside the water. So this is hypohydrophyly and this is epihydrophyly. So where it happens on the surface of water, alright. Yes, so this is your Zostria, you can say. Okay, so the names of the slides have been interchanged. So this is still your water pollination. Alright, so the sea grasses, the female flower is submerged inside the water. So your Zostria is your sea grass, alright. So here the the pollination happens inside the surface of water. So this is the height of the plant, okay. So it is deep inside the water. So here water currents carry the male gametes. So the pollen grains are released inside water. The pollen grains are long, ribbon-like and carried passively inside water to reach the stigma. So in the water pollinated species, so this is very important point. So in water pollinated pollen grains, the pollen grains are covered by a mucilaginous covering, so that protects them from wetting. Of course, your pollen grain has the outer sporopollenin, so surrounding that you have a thin mucilaginous covering so that they do not get wet, because this is water, right? So that is the reason. So these are the two types of pollinating agents that I have discussed today. One is your wind and the other one is your water. The third one is your biotic agent, that is your animal. So we will talk about it in the next class. Alright, so both water and wind pollinated flowers, they are not colorful and they do not produce any nectar. So this is also one criteria for your abiotically pollinated plants. Fine. So with this I will stop, because animal pollinated is a bit long and I want a continuous lecture for that. So this chapter is to be continued. So we will talk about animal pollination, that is pollination by animals followed by double fertilization and post-fertilization events in the next class. So this chapter is to be continued. I hope you understood whatever I taught today. Yes, so if you genuinely liked my way of teaching, so please do like, share and subscribe and stay tuned for more lectures from me. Alright, so thank you so much for watching my lecture and I'm waiting to meet you very soon. So take care and have a nice day ahead. Bye-bye.